Hello everyone. So I'm going to start chapter 9, and this chapter talks about microbial growth. It will talk about how actually bacteria grow, what are the process of bacterial cell division, how much time it requires to divide. Then it talks about different types of media, counting procedure, like how are you going to quantify bacteria. So that's all about this chapter. So first thing, that how bacteria actually grow. The process, name of bacterial growth process, or the division, how actually bacteria divides into two, that's called fission or binary fission. It's fission, the word fission we have studied during chapter 2. The word fission means when the parent cell splits into two cells. Okay. Binary means so basically from one cell we are going to get two cells. Bacteria actually divides by the process of binary fission. So the process, what happens actually first, the parent cell grow at certain level, means it actually stored all the protein and enzyme to divide its DNA or genetic materials. Then it divides the genetic material into two. Basically, it replicates its chromosome. And then that two sets of chromosome goes to the poles, both the poles of the cell, and then ultimately in between the chromosome materials, the division, or you can say the plasma membrane and cell wall actually originates, and that's how two cells get ultimately, they distribute the cytoplasm division occurs, and ultimately they completely separate from each other. So first step is chromosome division, and then the cytoplasm division. So after parent replicate its chromosome, then both the chromosome goes towards opposite pole. Okay, they start moving towards opposite pole. Then at the center, one special type of protein accumulates, and this protein is called FITZ protein. Okay, at first that protein granules, they makes a ring-like structure, and then slowly all those protein granules, they starts become accumulating, and solidification start, and then that feeds the ring actually start pulling the plasma membrane, okay, from both the sides, and that's how they make the septum or the divisome. It's called divisome or the septum. And ultimately, when the cytoplasm get completely separated by the divisome, then we call that parent cells are actually divided into two, or you can say that binary fission is completed. So that's the process of binary fission. Generation time. So every time bacteria, from one bacteria we actually get two bacteria. So each new fission, means each new binary fission cycle or generation increases the population by a factor of 2. Okay, and every time from one bacteria we get 2 to 4, so we can tell that population increases by factor 2, and every time it is by factor 2. So that's why this growth pattern is called exponential growth pattern. So basically it steadily grows, okay, and every time by the multiple of number 2. So that's why that's the exponential growth. Now the time, how much time it takes to divide, that depends on what type of bacteria. Like E. coli it's only take 20 to 25 minutes, E. coli. Then on the other hand, that Mycobacterium tuberculosis or smegmatis does not take that much time, but Mycobacterium tuberculosis, it takes at least 9-10 days to divide. So it depends how much time it will take. That depends on specific bacteria. So how we actually calculate the number of cells, okay? Um, so size of a population, how are we going to calculate the size of a population? So let's say we started with 2 bacteria, okay? And then after 1 hour or maybe after 2 hours, how many bacteria would be there? How are you going to calculate that? So to do that, we have a formula, okay? It is NF equals Ni multiplied by 2 to the power n. Okay, so basically, let's say it's urine infection, okay, of a patient, and at the beginning when we counted, when we made a streaking plate and we counted that there were maybe 4 bacteria were there at the beginning. So we started with 4 bacteria. So which stage the bacteria belongs in its growth phase, basically, like uh, to find out the status of the infection, okay, how old the infection is. To find out that, you must need to find out the number of bacteria after a certain period of time, okay. So basically, to do that, we use this equation. So NF means the final number of bacteria, Ni means that's the initial number of bacteria, N is generation. Okay, so after how many generation we are going to uh, observe. So over here, so let's say we started with 1 cells, okay, so that means the initial number was 1. Okay. Now generation, maybe we want to go, or we want to take up the second generation. So the formula will be Ni means initial was 1. So 1 multiplied by 2 to the power n, and we are going by 2 generations. So basically 1 into 2 to the power 2. So 2 to the power 2 equals 4. 4 should be multiplied by 1. So after second generation, we are going to have only 4 cells. So the math is really easy, like to do the math. So we started with, so if this is the formula, so what would be the NF? Okay, so N equal, today I don't have my stylus, so it's getting very difficult. I don't have that Wacom tablet, usually what I use to write down. So let's say N equal 2. So basically we are going for 2 generation, okay? And in I is, we started with, let's say, three bacteria, okay? Then what would be NF? Then NF should be 3 multiplied by, so formula is 2 to the power n. So 2 and n equals 2, so 2. So basically then 3 multiplied by 2 to the power 4 means it is going to give me 12. So that's the answer. So that's how you have to do the math. Make sure you memorize the formula. I'm probably going to give one question from this formula. So this is for you. Try to do the math and let me know what's the answer, which one is the answer. Okay, okay, let me do this for you. So if you have 100 bacteria, so the formula is NF equal, so NF equals NI, means initial number, and multiplied by 2 to the power n. Okay, so we have to find out n. F means final number. So if you have 100 bacteria, so we started with 100 bacteria, so that means Ni equals 100. Okay, with the doubling time 20 minutes. So basically that generation time is 20. So every 20 minutes they divide in the media. How many cells would be there in 2 hours? So this group of bacteria, they take 20 minutes for one time to divide basically. So 2 hours, so 2 hours means 120 minutes, right? Okay, I can't write with the mouse. Okay, 120 minutes. Okay. Now it takes 20 minutes to divide. So how many times it will divide within 120 minutes? And that would be our generation time. So basically n should be n equals 100, oh, I cannot write, 120 divided by 20, and that gives me 6. So basically 6 times they are going to divide. So that means our generation time n is 6 generations. So now if we do the math, if we put the number over here, so we started with 100 bacteria. So 100 bacteria will be here, multiplied with 2 to the power n, and we found out n equals 6. Okay, so 2 to 4, 4 to 8, 8 to 16, 32, 64, right? 2 to the power 6, 2 to 4, 8, 16, 32, 64. So 100 multiplied by 64, that gives me 6,400. That's the right answer. Batch culture. Okay, so bacteria, when we supply a limited amount of nutrient media, and we after that we actually do not provide any extra media, then we can see there are very specific 4 stages of bacterial life. Okay, so in lab, whatever the culture we use and whatever the process we use, that's called batch culture. Okay, so technique used to grow microorganisms or salts, limited supply of nutrients for growth is provided. When these are used up or some other factor becomes limiting, the culture declines. After a certain period of time, bacteria won't be able to grow, okay? Because all the sugar, all the proteins, they are going to metabolize, okay? And not only that, so they are doing the metabolism, okay? Means they are eating, they are growing. So at the same time, all the waste product will accumulate inside the media, like what you when you grow the bacteria at lab, like inside the liquid media, inside a tube or inside the agar plate, what we actually do, that's actually batch culture. So what we do, we put all the nutrients, we actually estimate, like let's say that we want to save the bacteria for 1 month. So we estimate that much amount of food we provide, okay, inside that liquid media, TSA plate or TSB broth media. And we add maybe 100 bacteria inside that media. So bacteria start multiplying, okay? Means they start utilizing the sugar and then protein of the media. So basically that's the food nutrient media. So they start breaking down all those nutrient media. When they're metabolizing those or breaking down all those nutrient media, at the same time they are going to produce waste material, right? So all those waste material inside the tube or plate, we are not going to clean that. So everything will be there on the plate or inside the tube, basically inside the media. So ultimately at certain point of time, the waste product will be so much because bacteria is multiplying constantly, right? So the number of bacteria is growing, and the amount of food particles and nutrient media is going down. So number is going high, amounts of nutrient is going down. So when this is the situation, amount of waste material is also really high. So high waste material, so you can imagine like let's say high amount of carbon dioxide can grow on inside that media. And what will happen? Bacteria cannot breathe if they need oxygen for their metabolism. So bacteria will actually die. At certain point of time, bacteria will die. So when the culture media, we make the culture media like that way, like we just supply nutrient media one time and we don't replenish that and we don't clean the environment, that's the batch culture, okay? um, and inside that batch culture, we actually intentionally we make batch culture, because inside the batch culture, population displays a predictable pattern, okay? And by seeing that pattern, we can make a growth curve, okay? So that's the purpose. And this is very important, this batch culture, because batch culture actually represent our body inside the cell, okay? Inside the body, like just try to consider that if it is a human cell, okay? And that inside that cell that cell get infected with the bacteria. So the cell has capacity, how many bacteria and for how many days it can carry all those bacteria. After that, what will happen? All the nutrients, sugar, everything will be deployed, with the by the bacteria, uh, and the cell will be filled up with waste material, nitrogen, carbon dioxide, the poisonous gas, and acid, means the product of the metabolites will accumulate inside the cell, right? And after certain period of time, bacteria will not survive. So that actually represent batch culture, what we do in lab, inside the laboratory. So what happens inside that batch culture? Bacteria actually show um, different phase. There are 4 phases of bacterial life, or you can say bacterial growth, that we can observe in a batch culture. Okay, so by seeing the batch culture means when we actually like let's say it's a blood, human blood. So by just observing the situation of the cell, we actually can recognize the growth phase of the bacteria, and based on that we can treat the bacteria, okay? So I will explain that in detail shortly. So inside the batch media, there are 4 phases: lag, exponential growth phase, stationary growth phase, and death phase. So basically what happens, so the uh, um, uh, growth curve is actually looks like that. So first is the lag phase, this one, okay? Uh, so lag phase, you can see that it's a very straight line, okay? versus log phase, it's exponentially increasing phase. Then again stationary phase, that's again a straight line, and then death or decline phase when the curve uh, goes down. Okay, so these are the four phases. So what's the meaning of this four phase? So the lag phase is this one. What happened during this phase? It's a straight line, means the number of bacteria is not growing. Number is remaining same. So if this bacteria started with 100, let's say that 100 bacteria was there at the beginning, over here, this after this much time, still the number of bacteria remains same. So what's the reason of lag phase? Because when you transfer the bacteria, when you inoculum a new media, you already learned, right, that liquid to liquid transfer and solid to solid transfer. So when you transfer the bacteria, very little amount of bacteria you transfer into a sterile media. That's a nutrient media. Okay, so at the beginning, it takes time for the bacteria to adjust in a new solution. Basically, the new temperature, new environment, new nutrient media, that different oxygen supply, different humidity supply. So all together, it takes for the bacteria a little bit of time to settle down. Okay, not only that, during this period of time, during the lag phase of time, um, they actually they start eating, they start metabolizing. Okay, but they start storing all the protein enzyme, so that also requires a little bit of time. Okay, so they grow in size, but they don't grow in number. Okay, that's the lag phase. So they grow in number. They store all the protein enzyme required to replicate the sets of chromosome, and then from here they start doing that binary fission. And when they start multiplying, then every time it grows the multiple of 2. So that's a exponential increase in number. We can easily calculate and we can estimate. Okay, so oh, where is my, what happened? Okay, sorry, I lost my PowerPoint. Anyway, so that's the uh, log phase. So lag phase, that's the number one phase. During that time, there is no division of cell. That's the time when bacteria actually settle down inside the new media. They start accumulating all the protein enzyme to grow or to divide. So that's the lag phase. But there is no increase in number of living bacterial cells. Now, log phase, when bacteria start multiplying by the binary fission, and at the next few minutes or few hours or few days, it will continue to grow exponentially. Okay, until the nutrients, they have the supply of nutrients media, they will exponentially grow. Now the third stage is the stationary phase. It looks like they reached a plateau, and it's again the steady number, because at this time, so they started from here to here. So you can understand. So 100 to the power 2. So every time they are multiplying by the number of 2. So over here, so many bacteria, they increased in number, so much number bacteria are there. So what happened, if you compare the number and the amount of nutrients at this moment, the amount of nutrients are less than the need. Okay, so lots of bacteria. So over here, the strong bacteria, they will survive. So survival of the fittest. Okay, so the bacteria, um, few bacteria, they will survive, but most of the bacteria, because of the absence of nutrients, supply of oxygen and humidity, they will start dying. So at this moment, rate of cell division and the rate of death are equal. How many bacteria um, are are um, multiplying, that number equals the number of death. Okay, and so that's why it's a steady state, and it is called stationary phase. Now at this moment, like over here, maybe there was 1% nutrients were available, but at this moment here 0% nutrients are available. And not only that, huge amount of waste material, excreta, deposited on the inside the media. So bacteria will die um, at exponential rate. So that's why over here they increased at exponential rate, but over here the number of bacteria is going to go down exponentially. Okay, so that's the death or decline phase. Now, inside the batch media, we actually can observe all these four stages. Knowing knowing the stage, you can actually diagnose the severity of the disease. Okay, like when it's lag phase, that number is not growing, they didn't start dividing. So if you apply antibiotic at this moment at lag phase, um, to destroy the cell membrane, cell wall, then they are not actually, that will not actually going to work. Okay, during that time, when they're dividing, when they're multiplying, then if you apply antibiotic, that will work. Again, if it's on the stationary, if the patient, inside the patient's body, the bacteria is in a stationary phase or death or decline phase, so this period, when it's a number 3 or number 4 phase, unnecessarily giving the antibiotic or to kill the bacteria, it does not make any sense, right? Because anyway bacteria, they are going to die. Okay, so why are you going to give extra medications that has side effects? So basically that batch culture, what we use in lab, that actually represent the um, cellular environment of our body. Okay, so that's the and inside that batch culture, we have, these are the four um, mm, stages of life of bacterial growth we can see. So I already talked about all this. So potential importance of the growth curve. Okay, um, so why it's so important? So implications in microbial control, you can control that, right? So basically, when the bacteria they are on log phase or lag phase, it's very important to control them. Okay, for infection, for food microbiology and culture technology, growth patterns in microorganism can account for the stages of infection. Understanding the stages of cell growth is crucial for working with the cultures. Okay, I hope you got the point why it's so important. Now, sustaining microbial growth. So we talked about the batch culture that represent the cellular cell environment of our body, where we don't resupply the food, we don't clean up, we cannot clean up the environment. Okay, but chemostat that's the culture vessel where we actually resupply the food material, and we can clean the bacterial environment, so that we can effectively allow bacteria to grow or to survive for a long period of time. Okay, so if that's the purpose, like when we want to make vaccine, or let's say if it's a research purpose, then we grow bacteria in a chemostat. Inside a chemostat, we have two extra tubes, where one tube that actually resupply the food, and another tube, with the help of another tube, we actually clean the environment, like all of their stool or excreta or the metabolic products, extra gas, extra acid, poisonous elements, we can clean, so that bacteria can sustain for a long period of time. And that machine is the chemostat. Measuring growth, how we actually count the number of bacteria. So two different methods. Okay, one is direct methods and indirect methods. Direct methods count individual cells. Okay, are more accurate, but takes a lot longer. So that's the exact counting of bacterial cells, but it takes time. Indirect methods, we cannot actually count the exact number, but what we can do, we can estimate the number based on the turbidity or the dry weight. So how much cloudy it is, based on that, or maybe we can take the weight, we can measure the weight, like just the solution plus after the growth, how much it is. Basically, based on the differences in dry weight, we can we can estimate the number. Okay, this is less accurate, but it's very quick. So direct total cell count, when we are talking about the direct total cell count, or indirect count, so basically over here we are talking about the viable count. Viable count means when bacteria are alive. Okay, so basically how many living bacteria are there, that's what we are going to count. Okay, that's important for the water quality, for the safety, for the soil, let's say for drinking water. So that's the viable cell count. The word viable means living bacteria, okay, living cells. So let me write it down. It's called, oh, I cannot write. So it's called viable cell count, means how many living bacteria are there. So over here, one way is using Petrop-Hauser chamber. It's a slide, microscope slide, but it's a special type of slide. At the center of the slide, it is a little bit depressed, the central part of the slide, and it is calibrated. Okay, it has grid, and all the grids are measured. Okay, by estimating the volume of that area and how many bacteria are there within that volume. By counting that, we can actually estimate the total number of cells. Like let's say so this is the center of the slide. Okay, so this is the gridded area. So like let's say this, it looks like that. So let's say we have 2, 3, 4, 5 bacteria, 6 bacteria, let's say 6 bacteria within this 1, 2, 3, 4, 5, 6 boxes are there. Okay, so within 6 boxes we have how many? 1, 2, 3, 4, 5, 6 bacteria, 6 living bacteria, or 6 viable bacteria are there. Okay, now what's the area of one box? So let's say from here to here, this much is 2 centimeter or 2 millimeter, let's say. So this much is 2 millimeter, this much is 2 millimeter. Okay, so that's the area. And if we place a cover slip, the height that is 2 millimeter. So um, that so if you multiply 2 to 2, that will give you the volume of one box. Okay, so basically over here I can see for 8 millimeter cube. Okay, so from this math I can get 8 millimeter cube. So that's the volume. Okay, so that's the volume of one box. Volume. Why we are going behind volume? Because that much water is there. Okay, so let's say it's a drinking water, and we are going to we are trying to find out that how many bacteria are there per 100 ml of drinking water. So that's what our target is. So if over here, so one box, small box has 8 millimeter volume, means 8 uh, millimeter, that much water we can store over there. Now, so 6 boxes are there. So 8 multiplied by 6, so 48 millimeter cube, that's the total area. So within that area, we have 6 specimens. So then we can do the math. So that's how we actually can find out, we can estimate the number. Why I'm saying it's estimation? Because sometimes, you know what happened, that under the slide, under the microscope, this slide, like over here, you can see the dot-like structure, very small dot-like structure, the bacteria. Not all the time all bacteria are alive. Okay, but under the microscope and within this Petra-Forscher chamber, you really cannot differentiate between dead cells and living cells. So sometimes it's only a dart, but it looks like bacteria. So all those things can happen. So it's not exact count, but it's pretty close. So that's why it's a direct count. So that's the math. Another ways to count, it's called Coulter counter. This Coulter counter, it's actually check the density. So basically it has inside the counter, inside the chamber, there are actually 2 chambers are there. One is large chamber like this one, and this is the small chamber. So you can see that U-shaped structure, that's a small chamber. There should be there must be one opening between these 2 chambers. Okay, and we place 2 electrodes, and both these chambers are filled up with electrolytes, in salt water. Okay, so when we run the electricity, what happened, that salt waters, that electrolytes, they start moving, right? So over here, at the side of that opening, or you can say the between the large and small chamber, we actually place a detector. It's called a resonator. So and it is actually aligned with the size of the electrolyte particles. So different than that electrolyte particles, like if it is a bacteria, this resonator, it can detect. So so basically because of the electricity, there is a movement of water and the electrolytes, right, between these 2 channels. So instead of salt particles or electrolyte particles, if the bacteria moves through this hole, then, or through this opening, then that resonator actually can count. It can detect. Okay, so by counting that number, we can actually estimate the number of bacteria. So that's the Coulter counter. method. Same thing. um, um, Another one is colony forming units, how many colony are there, that what we actually do when we make the streak plate, right? Liquid media, you really cannot count the colony, but in a solid media you can see the colony, right? Visible colony, and you can count. And remember, in lab I told that one colony represent one bacteria. Okay, so if I have 1, 2, 3, 4, 5, 6 colonies on the plate, that means at the beginning 6 bacteria were there. Okay, so that's how we can count. And this is called colony forming units. Okay, so um, um, let's say if we want to count that in 1 milli of water how many bacteria are there, then what we have to do, we have to take 1 ml water on the plate, okay, nutrient media, and then we have to allow the bacteria to grow, and then we can count the number of colony, and that would be the colony forming unit per liter of water, and that's how we actually count. But the thing is, I told that 1 liter of water, so we cannot really take that much water in a plate, right? So basically we go by very diluted form, and that's the milliliter, and that's the unit. So CFU per ml, that's what we count. Um, but there is a range. We only can count if there are 30 to 300 colonies on the plate. Okay, below 30, that's negligible. So on a plate, if there are less than 30 colonies, that is negligible. And at the same time, if there are more than 300 colonies, then that is also negligible. So our range is 30 to 300 colonies. Two methods are there, pour plate and spread plate. You both we used the spread plate technique. So basically this is our media, and what we did, we took the bacterial mixture, or you can say the water or whatever you want to experiment, then you spread it properly and allow uh, the basically incubate the plate, and then next day you can count. That's the spread plate technique, one way. In lab we usually use that spread plate technique. Pour plate technique is instead of directly take the water or bacterial mixture onto the plate, we actually warm up the agar, liquid agar, and we add up that water or that sample. Okay, and then we pour the plate, and then we shake the plate in a manner, and then we allow it to grow. So that's the pour plate method. These are the two methods. Now, so probable count means when we are going behind the colony forming unit, like how many bacterial colony are there. So we can do that from pour plate, or we can do that from spread plate technique. Both are same. So basically, by counting the bacterial colony uh, on the number of plate. Okay, but the thing is, remember that I told that the range is 30 to 300 colony. Okay, so if your water sample or soil sample is too polluted, okay, um, uh, then inside the plate there will be more than 300 bacteria. Then you really cannot count. Okay, that will not make any sense. You won't be able to count. So what we do before we make that spread plate or pour plate technique, we actually serially dilute the solution. Okay, like let's say we want to check the Lake Alatuna pond water, okay, water quality. So if it's near that filtration unit, like where everything the county is dumping, if that area, and if the company is not doing properly, so that area could be polluted very much. So if we directly take that water and if we want to make the plate, then it will be too many bacteria, we cannot count. Okay, so what we do actually, we actually serially dilute that water. So how we do, so we actually for based on the pollution level actually, maybe 3 4 5 or 4 times dilute you have to do. So each tube will carry 9 ml of water. That's just the distilled water to dilute. Okay, and this is your original sample. That's, let's say that's your pond water. So what we are going to do, or maybe that's your drinking water, which one you are going to take or taste. So we are going to take 1 ml of original sample water into 9 ml of distilled water. Okay, so in this tube we had 9 ml of distilled water. Now we added 1 ml of sample water. So altogether we have 10 ml. Okay, liquid in this tube, but within the 10 ml tube, original sample was, oh my phone is, original sample was only 1 ml. Okay, so then we can represent it as 1:10, right? So original sample 1 portion, but all together 10 portions. So out of 10 portion or 10 ml water, we have original sample 1 ml. Okay, now for the next tube, what we do, so next tube also has 9 ml distilled water. Okay, now this time instead of taking original sample, we are actually going to grab 1 milli from the first tube. So basically we are trying to dilute the water, right? So this time we are going to grab 1 ml of sample from this tube. So over here, this original sample is already diluted 10 times, right? So when we grab 1 ml from here to here, so altogether this tube now it has 10 ml of water. But the dilution level is 10 multiplied by 10. So basically it gives me 1 out of 100. Okay, so within 100 ml of water sample, we have only 1 ml original sample. That's how basically we have filtered the water 100th time. Now, if we continue, so this tube will give you 1:1000, then 10,000, then 100,000. So based on the pollution level, if you feel it's really polluted, then you have to dilute multiple times. But if it is drinking water and you think it's not that much polluted, so 1 or 2 times, so maybe up to 1,000 should be fine. So serially we dilute the concentration. Now, what we do from this tube, we collect, half ml. This time not 1 ml. Sample from this tube onto plate, and we do the spread plate technique. Over here again, half ml. So half ml, and then we make the corresponding plates based on the solute concentration, and then we actually incubate all the plates. So over here, the sample concentration was only 1:10, 1 out of 10, right? So too many bacteria will go over here. The next one, yes, over here also it will be too many, because the concentration is only out of 101. So over here, out of 1,000, we first 3 probably we cannot count. Maybe over here it's 50 colonies, because we diluted, right, serially, and maybe over here 2 colonies. So these 2 would be countable. if it is a pond water. But if it is drinking water, probably the second or third plates we can count. So this process is called serial dilution, and very, very important. Before we make this pour plate or spread plate, we actually do the serial dilution for the original sample. So this is the way we check the water quality. This is the way we check the drinking water quality. We check the soil quality, anything, any pollutant, disinfectant, everything. When we want to see the effectiveness, we actually go by this. Another way to quantify the bacteria, to count the number, it's called most probable number. It's very, very important, MPN. It's the statistical way to count the number. So in this way, there are very specific bacteria. It's called coliform bacteria. Okay, so the name of the bacteria is I cannot write. It's called coliform bacteria. So coliform bacteria means they remain inside our gut. Okay. Um, so if inside your drinking water, um, if we have coliform bacteria, that means that water is polluted, because uh, coliform actually gut bacteria means that came from your gut. So you can understand that that water is infected with the sewer system or water, right? So the coliform presence of coliform inside a drinking water or any water, that represent the infection. Okay, and that infection actually comes from the sewage system, um, or from the animal products, on animal excreta, you can say. So, and this coliform, they have another characteristics. They can ferment lactose. Okay, so they ferment lactose. Means lactose one sugar. So basically, not all bacteria can ferment all types of sugar. Okay, if they have enzyme, specific enzyme, then only they can ferment that sugar. So coliform bacteria, they have lactase enzyme, so they can ferment lactose, okay, sugar. So what we do, we actually take 3 sets of 5 tubes, okay, and each 5 tube has lactose nutrient broth, okay, and we take 3 tubes, 3 sets. So what we do, the first set, we actually inoculate the first 5 tubes or first set of lactose tube with 10 milli of sample. Okay, then the second set we inoculate with 1 milli. So basically serially we are diluting. Okay, so 3 different diluted sample we are adding. So over here 0.1, and then we allow bacteria to grow. If coliform bacteria is there, then they are going to ferment that lactose, and that media has special type of dye. If bacteria can ferment lactose, then that pink color will change into yellow color. Okay, and by seeing that color differences and counting how many tubes are showing positive result, we have a chart, statistical chart like this, and then we can tally, and that's how we can find out the most probable number. So over here we can see the first set, like when it was 10 ml, very strong, that 5 positive tube, over here 2. So our number is 5, 2, and 0. Over here we don't see any. That when we inoculated the lactose tube with 0.1 ml sample, then there was no positive result. So our result is 5, 2, and 0. Now if we tally with the chart, so we can see the 5, then 2, 0, that gives us 49. Okay, so that means in that sample, per 100 ml water was carrying 49 coliform bacteria. Okay, so that's how we go, we check that most probable number. Indirect and total count mostly by the as I told, that based on the cloudiness. Okay, we do so basically by seeing the turbidity, we have special machine, it's called spectrophotometer over here. So what we do, we pass this one special wavelength of light through the sample. Okay, so if how many, basically how many light rays can pass through, um, um, that the detector will actually count that number. Okay, so if it is too concentrated, so light rays will go and will struck back, right? Because they will hit the bacterial cells, and they won't be able to go pass through the solution, right? So if they hit a bacterial cells, they will return. Okay, but if they don't hit the bacterial cells, then they will pass through the solution, and they will touch the detector. So and detector will count that number. Okay, how many light rays actually reached to the detector. So that's the machine detector, that's the spectrophotometer, and it will count that number, and that's how we, so basically based on the turbidity, we can count the number. Biofilm, so how to quantify bacteria, we are done. Okay, now over here we will talk about a little bit about the media and bacterial growth. Biofilm, that's what is biofilm. Biofilm is actually a colony of so many different types of microorganisms. Biofilm does not means it's only bacterial colony. You can find out fungi, protozoa, viruses, lots of different microorganisms together. They make a colony. Okay, and then when they make the biofilm, they are actually going to change genetically. They behave like one single unit. Okay, so let's say inside the biofilm, so inside your teeth, maybe Pseudomonas, E. coli, Streptococcus, Staphylococcus, all these bacteria are there. But when they make, when it solidify, when they make a biofilm, when it's a biofilm, means some genetic changes occur inside. They are not just the E. coli or Streptococcus or Staphylococcus or Pseudomonas. You cannot treat biofilm with that specific antibiotic. It will not work because it's completely different identity. Okay, so that's the biofilm, and it's very difficult to kill the biofilm. So that's why when plaque grows inside your teeth, that's not healthy. Okay, you really should not allow to grow that plaque inside your mouth, because if it becomes loose and it can go inside the blood circulatory system, means if it can moves through the artery or veins, what will happen? It can block the artery, veins, or even the heart. So it's very risky. So how actually they make biofilm? So first, that floating microorganism we cannot see, that's called zooplankton. Okay, so all the zooplankton slowly they need to settle down. There should be a surface. They only can make biofilm if there is a surface, solid surface. If there is no surface, the attachment side, then they cannot make the biofilm. So first they settle down. Okay, and once they settle down, they become sessile. what they do, they start secreting some type of organic materials, okay, and that actually attract other organisms to go over there together. Now, at certain point, um, basically at first stage they become sessile, but it does not mean that that's the permanent attachment, okay. Sometimes a few of the microorganisms, they can go back to their original life, okay, because they only start secreting organic molecules as food. But at certain point, they become tightly attached on the surface. Not only that, they start secreting one special type of polymeric substance. It's called extracellular matrix, or EPS, extra EPS. Okay, that EPS, when once the microorganism starts secreting that EPS, extra polymeric substance, it's very sticky, large carbohydrate, um, then they cannot go back to the plankton form again. Then their genetical configuration has been changed, okay, at this moment. um, and that has a smell. Other bacteria attracts, they accumulate, and ultimately they make a colony. So that's the biofilm. Interestingly, inside they have so many different, they have water channels. It's so hi-fi, that colony. They make channels so that water can come in. They make channels for air circulation, so oxygen can come in, and carbon dioxide can go out. So all these channels are there. They do quorum sensing, I mean, they communicate with each other. One organism communicate with another organism. Like, let's say we don't have enough food, so uh, um, some of the member needs to die. They actually transferred that order. So few people, they're the martyr, they die. So all those things happen. How scientists still are working on this quorum sensing and biofilm. Okay, it's not clear at all. We don't know everything about this quorum sensing and biofilm, but scientists believe that chemical induction and I'm not sure about. I really don't know exactly what happened, but it's because of the different types of chemical concentration. They can actually interact with each other. That much I know. So oxygen requirements, different type of bacteria there. We already worked on this lab. So obligatorab, so they grow only at the surface of the broth, okay, because they need high amount of oxygen. Obligate anaerob means they don't need oxygen, and their obligate means they will grow at the bottom, okay. So in a test tube, or like a tube like this one, so as you go to the bottom of the tube, oxygen concentration goes down. So obligated, they will grow at the top surface of the media, growth media. Obligate anaerobe, they will grow at the bottom of the tube. Facultative on anaerobes, they grow throughout the tube. Aerotolerant anaerobes, they really don't need oxygen, but they can tolerate. So they will also grow throughout the tube, and the tube will look cloudy. Microaerophils, they need very little amount of oxygen, but they cannot tolerate if there is too much oxygen. So instead of growing on the surface, they actually grow little below the surface, where oxygen availability less than the surface. So that's the microaerophils. pH requirements, optimum, anything like optimum oxygen. So the word optimum represents the most favorable environment, okay, you can say. So when, if it is optimum pH, means the pH level which is most favorable for the growth of the organism. That's the optimum growth pH, okay. Based on the pH requirement, means acidity or alkalinity, neutrophils, they're neutral, so they grow best around pH 6 to 8. Acidophiles grow best below 6 actually. Alkaliphiles, they grow best above 7. Acidotolerant, basically they are neutrophils, but they can tolerate the acidity. So how are we going to find out the optimum one? So from the graph, it's very easy to find out. So we go by, from the peak, we go at the center, okay? So for the acidophiles, means they can tolerate acidity, high acidity, so that their range is from 1 to 5.5. So if the media pH varies from 1 to 5.5, that acidophile, they can grow. But what's the optimum temperature? So from point, if we draw a line at the center, so basically that hits number 3. So that's the optimum pH for acidophiles. Exactly same way, our optimum pH for neutrophile is 7. Alkanophile, so it's 9.5. Okay, so that's how we can find out the optimum temperature, optimum pH, or anything optimum condition. So psychrophile, based on the temperature, we have psychrophile, mesophiles, thermophiles, hyperthermophile. Psychrophiles, they can tolerate -5 to 20 degree temperature. Okay, and then you can understand that what would be the optimum temperature. Mesophiles, mesophiles, or that's most human pathogens, because that represent our body temperature. Okay, mesophiles, they can grow from 15 to 45 degree temperature. Okay, um, but their optimum temperature is almost 30 to 35 degrees centigrade. So thermophiles, they can grow from 40 to 80. Optimum temperature should be 60. Hyperthermophiles, they can tolerate huge, high temperature, more than 100, so almost 110 degree centigrade temperature. And their optimum temperature, that 85, 90 degree temperature, when they grow comfortably. Osmotic pressure based of solute concentration, like the situation is hypotonic or hypertonic. We have two different types. Osmophiles live in habitats with high solute concentration. Any high solute concentration, high sugar, high salt, they are called osmophiles. If it is high salt concentration, then they are called halophiles. Like Staphylococcus epidermidis, they are the halophiles bacteria, because they can tolerate high salt concentration, and that's why they live on your skin. Now we are going to talk about media, growth media basically. So first one is general purpose media. So basically designed to grow a broad spectrum of microbes. Okay, so many different variety of microbes we can grow in this media. Any nutrient target, like what we use for the, TSA and TSB plates, that's the general purpose media. Specialized media, like if we want to observe the bacterial behavior, like pH indicator, is it a thermophile or hyperthermophile bacteria, and if we add any extra dye or extra pH indicator inside the media, then that's the specialized media. Okay, or if we want to observe that antibiotic gene, presence or absence of antibiotic gene, so if we add that antibiotic, like let's say it's just regular growth media. Okay, and with that, if we add let's say streptomycin antibiotic, okay, and if we grow the bacteria and bacteria can grow, that means we can tell that, okay, so these bacteria, they are streptomycin resistant. Streptomycin will not work onto that bacteria. But if bacteria cannot grow, that means streptomycin still can work on to kill all those bacteria. Okay, so when we add that antibiotic with the general purpose media, our sterile growth media, that becomes a specialized media. So to make that specialized media, we have special purpose. Enriched media, this is for fastidious microbes like picky eaters. They need special type of food like Neisseria gonorrhea. For gonorrhea, they're responsible for gonorrhea. They must have chocolate agar. Chocolate agar means basically it's a lysed RBC, lysed RBC. So basically we break down the RBC and that gives a brown color, dark brown color, and it looks like chocolate. So that's why it's chocolate agar. So there are a few bacteria who need special type of food. Okay, so that's the enriched media, and those bacteria or those microbes are called fastidious microbes. Defined and or synthetic media means we know all the ingredients. Okay, um, each and every ingredients we know, and we also know the measurement. So basically, composition is very clear. Complex or non-synthetic media, we really not sure about the exact composition. At least one component should be unknown. Okay, so defined a synthetic media like what we make here at Georgia Highland, it will be same if Kennesaw State University make, okay? But the complex and non-synthetic media, it would be completely different based on, you know, um, where and how you are going to make. Another important media is selective media. Selective media, they actually select a group of bacteria to grow, okay? Um, So basically what they do, they control the growth of one group of bacteria and then they allow another bacteria to grow. Like let's say EMB, eosin methylene blue agar. This media, EMB agar media, they're selective for gram-negative bacteria. Means they can allow gram-negative bacteria to grow. Any gram-negative bacteria you can grow on EMB plate. But if it is a gram-positive bacteria, you cannot grow gram-positive bacteria on EMB plate, because this media, that eosin methylene blue agar or EMB agar, it will actually block or control or inhibit the growth of gram-positive bacteria. Okay, so it actually selects selective against gram-positive bacteria, but selective for gram-negative bacteria. Mannitol salt agar only halophilic bacteria, means the bacteria who loves high amount of salt, they can grow. McConkey are selective for gram-negative bacteria. Only gram-negative bacteria can grow. P-plate, phenyl ethyl alcohol agar, only gram-positive bacteria can grow. So these are like you want to check very quickly. You want to check that, okay, which group of bacteria, gram-positive or gram-negative bacteria. So what you have to do, instead of regular plate, regular sterile nutrient media, you can use this plate. Okay, so let's say you can choose McConkey agar plate, and you take the stool sample over there. So if you see that, okay, so after 24 hours, I can see the colony on that plate. That means inside that stool, there was gram-negative bacteria. So it was a gram-negative bacterial infection. So you can prescribe antibiotic for that gram-negative bacteria. Okay, so that's the purpose of that selective media. You can actually differentiate. Differential media, you actually can differentiate based on the color, shape of the colony, size of the colony. You can differentiate mostly the color, like EMB plate. As I told, that EMB is both selective media and differential media. it is selective for gram-negative bacteria. Okay, so only gram-negative bacteria can grow. Okay, positive they cannot grow. Now, this media we also can use if bacteria can ferment lactose or not. Okay, lactose positive or lactose negative. So this media has another dye that actually, so it can allow all gram-negative bacteria. So all gram-negative bacteria will grow. But if it is coliform bacteria, means they are lactose positive, they can ferment lactose, then what they are going to do, they will change the color. Okay, so that media at the beginning it was red. Okay, but if lactose positive bacteria are there, then it will convert the color into yellow. So next, if you see that that agar itself, the media actually has changed its color from red to yellow. That means you know that lactose bacteria are there, means the presence of coliform bacteria. Okay, so it actually basically it differentiate and shape, size, color, sometimes gas bubbles are there. So that's the differential media. So the differences between selective and differential media, very, very important. Selective media just select one group of bacteria and completely inhibit the growth of other group of bacteria. So only either gram-positive bacteria can grow or gram-negative bacteria can grow. Okay. Differential media, it will allow both gram-positive and gram-negative bacteria to grow, but it will show the different color or shape or size, and you can identify. So that's the difference between differential and selective media. Now there are lots of media that we use in our lab, like EMB plate, eosine methylene blue agar, EMB plate. That's both selective and differential media. We actually add all those dyes to make it both selective and differential. So this media will only allow gram-negative bacteria, and at the same time it will differentiate based on the lactose fermentation. Okay, so we can go much specific towards the bacterial identification. Okay, so I think we are done this chapter.